Welcome to the lecture for transcription, which is the first step in protein synthesis. To start off today, we're going to talk about the big picture of protein synthesis. What does it look like as a big picture? We know that it is a two-stage process where DNA is used to produce proteins which code for traits. Remember from yesterday, the DNA lies in the nucleus, and it's too large to leave the nucleus. And the DNA has the instructions to make the protein. But the proteins are made at the ribosome, which is not in the nucleus. It is in the cytoplasm of the cell. So we have to end up finding a way to get the code from the nucleus to the ribosome to create the protein. And so the big picture here is that we have to take DNA. We have to create an mRNA with the code of that protein from the DNA so that the mRNA can travel to the ribosome. Once the mRNA travels to the ribosome, it takes amino acids, those monomers of proteins, if you remember. Amino acids are the small subunits. They take those and they end up transferring them to connect to one another to create a protein. Once that protein is created, then that protein will express the trait, the gene that you're trying to express. So an example here. In your DNA, you might have a trait or a gene, the instructions to have brown eyes. So the DNA is going to have instructions to create a protein that's going to cause your eyes to become brown. That code is going to have to be first transcribed into mRNA so that we can get that code from the nucleus to take it to the ribosome. That code is then used to attach a specific sequence of amino acids together. Once they're together, they can fold into a protein. And once those proteins are created, then you're going to have enough of those brown proteins to show off the trait of having brown eyes. So there are two phases to protein synthesis. We're going to focus just on one of the phases today called transcription. Transcription is transferring information from DNA to mRNA so that it can leave the nucleus and travel to the ribosome. So once again, that DNA is not able to leave the nucleus because it's too big, and so that first step is going to be transferring that code into an mRNA molecule so it can leave the nucleus and travel to the ribosome. Then the second step is called translation. Translation is where tRNA is going to decode the message from the mRNA to carry amino acids to make the protein at the ribosome. And we're going to talk more about translation later. We're first going to talk about transcription. So once again, the purpose of transcription is to transfer the information from the DNA molecule into an mRNA molecule because it is small enough to leave the nucleus. Why do we have to do it? DNA has the code for the protein that needs to be made. Proteins are made at the ribosomes. DNA can't get to the ribosomes because it's too large to leave the nucleus because it's double-stranded, but RNA can leave the nucleus since it is a single strand. And so it has to be transcribed into that RNA molecule to physically leave the nucleus. So where does transcription take place? Transcription takes place inside the nucleus because that DNA is going to be copied into an mRNA molecule so that it can then leave. The enzyme needed to do transcription is the enzyme RNA polymerase. Once again, if you see that ASE at the end, you know it's an enzyme. RNA polymerase binds to DNA, unzips it, and then uses the one strand of DNA as a template to build a second strand of mRNA. So you can see here, this DNA molecule has been unwound and unzipped, and it's just using this strand of the DNA on the right. And it's going to create the code, the complement code of that sequence. So if you notice, this A is not an A on the first nitrogen base here on the RNA. It is actually U, because for RNA, A binds with U. This T binds with A. There's not another T here. So it's the complement of the DNA molecule to create the mRNA molecule. Once again, there are no thymines in mRNA, and so you're only going to see A, U, C, and G. So when you see an A on the DNA strand, the complement is going to be U for RNA. Now, what other type of polymerase did we talk about that did this job? This actually was the job of two different enzymes. If you remember, helicase is what unwound and unzipped the DNA. But then DNA polymerase did this job for DNA replication. And so RNA polymerase is very similar to DNA polymerase, except that it's creating an RNA polymer instead of a DNA polymer. So let's recall, what bases pair together in RNA? A pairs with U, U pairs with A, G pairs with C, and C pairs with G. So if we are in the nucleus and this is our DNA strand, we've already separated it and we're using the left side as our template, we're going to create our mRNA. So we have A, the complement for mRNA, is going to be U. G, the complement is going to be C. C, the complement is going to be G. The next one, T, the complement would be A. C, the complement would be G. And then A, the complement would be U again. Now, after that mRNA is created, then it's going to leave the nucleus to travel to the ribosome. So we're going to do some quick transcription practice here. Transcribe the following DNA strand into mRNA. And so I want you to take a moment. You can just do it on, then you're going to do this on the next page of your packet where it says transcription packet practice, and you're going to find the mRNA from that original DNA template. I want to remind you, it is the complement of those. And remember what base we do not see in RNA and what base we do see in RNA instead. Go ahead and pause this video to complete those 4, and then come back to see what the answers would be. So the first one here, the G is going to complement with C, C goes with G, G goes with C, T goes with A goes with U, because there's no T's in RNA, and then T goes with A. So that would be the answer to the first one. The second one here, A goes with U, because there's no thymine. A goes with U, T goes with A, C goes with G, T goes with C, and T goes with A. And then the remaining 2 are as follows. So just check your work and make sure that you're creating the mRNA strand from the DNA strand. So what's next? After transcription, that mRNA needs to leave the nucleus and go to the ribosome, so translation can happen. So we've already transcribed, created that mRNA, and now the mRNA is going to leave the nucleus to go to the ribosome. So once again, mRNA just leaving the nucleus to go to the ribosome. Okay. So from here, you're going to complete your transcription practice in your practice packet. It's just that front page of the packet you're going to complete for the rest of today.